From the slime-coating slugs to the saliva in our mouths, many slippery bodily fluids contain mucus. So how did this marvel of biology evolve? In mammals, the answer is many times, and often in a surprising way, according to a new study on proteins called mucins. These molecules have a variety of functions, but as a family, they are known as components of mucus, where they contribute to the substance's gooey consistency. Through a comparison of mucin genes in 49 mammal species, scientists identified 15 instances in which new mucins appear to have evolved through an additive process that transformed a non-mucin protein into a mucin. The scientists propose that each of these mucinization events began with a protein that wasn't a mucin. At some point, evolution tacked a new section onto this non-mucin base, one consisting of a short chain of building blocks called amino acids that are decorated with sugar molecules. Over time, this new region got duplicated, with multiple copies added on to elongate the protein even further, making it a mucin. The sugars coating these sections protrude outward like the bristles of a bottle brush, and they bestow mucins with the slimy property that's vital to many important tasks that these proteins carry out. The research will be published on August 26 in Science Advances. I don't think it was previously known that protein function can evolve this way, from a protein gaining repeated sequences. A protein that isn't a mucin becomes a mucin just by gaining repeats. This is an important way that evolution makes slime. The repeats we see in mucins are called PTS repeats for their high content of the amino acids proline, threonine and serine, and they aid mucins in their important biological functions that range from lubricating and protecting tissue surfaces to helping make our food slippery so that we can swallow. My lab has been studying mucins in saliva for the last 30 years, mostly because they protect teeth from decay and because they help balance the microbiota in the oral cavity. The intriguing evolution of an amazing life trait. I think this paper is really interesting, Gökçemen says. It's one of those times where we got lucky. We were studying saliva, and then we found something that's interesting and cool and decided to look into it. While studying saliva, the team noticed that a small salivary mucin in humans called MUC7 was not present in mice. The rodents did, however, have a similarly sized salivary mucin called MUC10. The scientists wanted to know, were these two proteins related from an evolutionary perspective? The answer was no, but what the research uncovered next was a surprise. While MUC10 did not appear to be related to MUC7, a protein found in human tears called PROL1 did share a portion of MUC10 structure. It gains the repeats that give it the mucin function, and it's now abundantly expressed in mouse and rat saliva. The scientists wondered whether other mucins might have formed the same way. They began to investigate and discovered multiple examples of the same phenomena. And this was with a pretty conservative look, Gökçemen says, noting that the study focused on one region of the genome in a few dozen mammal species. He calls slime an amazing life trait, and he's curious whether the same evolutionary mechanism might have driven the formation of some mucins in slugs, slime eels and other critters. More research is needed to find an answer. How new gene functions evolve is still a question we are asking today, says Pajic, a UB PhD student in biological sciences. Thus, we are adding to this discourse by providing evidence of a new mechanism, where gaining repeated sequences within a gene births a novel function. I think this could have even broader implications, both in understanding adaptive evolution and in possibly explaining certain disease-causing verses. And then, at the other end of the spectrum, maybe if this mechanism goes off the rails, happening too much, or in the wrong tissue, then maybe it can lead to disease like certain cancers or mucosal illnesses. The study on mucins demonstrates how long-time partnership between evolutionary bio. My team has been studying mucins for many decades, and my collaboration with Dr. Gokyaman has brought this research to a new level by revealing all these exciting novel insights into their evolutionary genetics, Roll says. At this advanced stage of my career, it is also immensely gratifying to see that the flame of scientific curiosity is being carried on by a new generation of young investigators like Petr Pajic. Additional research co-authors include Shichen Shen, Ph.D., postdoctoral associate, and Jun Ku, Ph.D., PR. May is now an assistant professor at the Icahn School of Medicine at Mount Sinai. Scientists who conducted the study are supported by the U.S. National Science Foundation, along with the National Institute of Dental and Craniofacial Research and National Cancer Institute, both part of the U.S. National Institutes of Health. Materials provided by University at Buffalo. Original written by Charlotte Hsu. Note: content may be edited for style and length. Petr Pajic, Shichen Shen, Jun Ku, Allison J. The evolution of mucus. How did we get all this slime? In mammals, proteins called mucins evolved, again and again, by co-opting non-mucin proteins in a surprising way, study suggests. Science Daily. In mammals, proteins called mucins evolved, again and again, by co-opting non-mucin proteins in a surprising way, study suggests. Science Daily. Retrieved August 26, 2022 from ift.tt/m91 University at Buffalo. The evolution of mucus. How did we get all this slime? In mammals, proteins called mucins evolved, again and again, by co-opting non-mucin proteins in a surprising way, study suggests. Science Daily. https://ift.tt per meter 91 WKRT. Accessed August 26, 2022. Thanks for watching. Please subscribe my channel for more videos.